Thank you very much, Dr. Kim. First of all, I would like to thank organized committee to invite this meeting. So today I want to talk about self-renewal of hematopoietic stem cells. As you know, hematopoietic, can you see my slide?
Yes, well, please go ahead.
Hematopoietic stem cell transplantation is one of the ideal methods for blood disorders. And from now, we are thinking about hematopoietic stem cell expansion obtained from healthy donors, or ES-iPS cell induction to hematopoietic stem cells, or some transformation from progenitor or endothelial cells to hematopoietic stem cells. So there are so many trials, but still it's not so realized. So today I want to talk about the self-fate decision and the self-renew of hematopoietic stem cell in fetal liver. And finally, I want to talk about the ex vivo stem cell expansion. As you may know very well, so hematopoietic stem cell fate decision is a self-renewal or differentiation. Self-renew means stem cell give rise to stem cells. So that's a birth event. On the other hand, differentiation is a death pathway because differentiating cell show the life limitation of lifespan. So if the self-renew is very high, we can see expansion of the hematopoietic stem cell system. On the other hand, self-renew is very low, we can see extinction of the system. So almost 40 years ago, we did paired daughter cell experiment under the instruction of the mentor Makio Ogawa. I picked up a single cell from blast colonies, and when single cell divided into two cells, we could manipulate and check the colony formation. As you can see in this slide, colony A shows the multi-lineage differentiation. On the other hand, colony B shows the only macrophage. So single cell, single mother-derived paired daughter cell shows a very different differentiation pathway. This is a scheme of the disparate differentiation paired daughter cell. For example, one colony is a macrophage, another one is a neutrophil, macrophage, mast cell, erythroid cell, and myeloid cell, multi-lineage. So there are so many combinations and asymmetric cell division. And this is interesting. Megakaryocyte versus multi-lineage, megakaryocyte versus neutrophil macrophage. So we believe that megakaryocyte biased differentiation is already observed here. But at this time, we could not see this kind of the paired daughter cells. One is a multi-lineage differentiation, and the partner is just a stem cell, single stem cells. So because we are treating just progenitors, so it's very difficult to see the self-renewal. So 30 years later, we tried again this PDC, paired daughter cell experiment. At this time, we can purify the hematopoietic stem cells by FACS, and we deposited single cell in microtiter well. And when the cells are divided, we analyzed the gene signature of each paired daughter cell using the qPCR array system. And collaborating with Ben MacArthur in University of Southampton, he is a mathematician, and so he analyzed the stem cell symmetric cell division using the PCA analysis. This is the region of the long-term hematopoietic stem cells, and this region is a progenitor region. So two paired daughter cells show the still they are stem cells. So this is a stem cell symmetrical division. And if the paired daughter cells show the progenitors, this is the progenitor symmetrical division. So differentiation division. And this is asymmetric division. This is a very arbitrary criteria. So at first, we compare the 4 weeks age mouse hematopoietic stem cells with 8 weeks age mouse hematopoietic stem cells. As you can see in this slide, in 4 weeks, hematopoietic stem cells show the stem cell division very much compared with control, compared with 8 weeks. And we add angiopoietin 1. This is a type 2 receptor ligand, can affect a little bit. So we can see some self-renewal division increase by the addition of the angiopoietin 1. Next, we try to check the polyethylene glycol PEG hydrogel effect. So we plated the single cell in the Petri dish coated with PEG hydrogel. So we can see some increase of the self-renew in PEG hydrogel dish compared with just a normal culture plate. So maybe some attachment, cell contact is very important for the stem cell self-renewal. And then again, we can see angiopoietin 1 effect to increase self-renewal. So we can conclude that cell fate is determined by probability of intrinsic and extrinsic effect. So age dependent, this is a very intrinsic, and niche dependent. So angiopoietin 1 or hydrogel presence or absence, which will affect the self-renewal activity. So very recently, we are analyzing the fetal liver hematopoiesis. So order of the stem cell is ordering of the stem cell is very opposite in the imaging. So usually hematopoietic stem cell produce LMP, EMP, or erythroid cells. However, in embryonic stage or developmental stage, at first we can see erythroid progenitors and EMP and lymphoid myeloid progenitor. And finally, we can see hematopoietic stem cells on day 10 mouse embryo. So there are so many research about the development of the hematopoietic stem cells in AGM. AGM is a very interesting mesodermal region, aorta gonadonephros. So cardiac system, artery system is very close to hematopoietic system. So we can see some endothelial cell produce hematopoietic cell like this. This is a type 2 monoclonal antibody staining. So we can see a two-way of the hematopoietic stem cell generation. These homogenic endothelial cells produce genuine hematopoietic stem cells or EMP, just progenitors. This difference is very important because EMP is not transplantable. Only HSC can be transplantable. So we would like to discriminate hematopoietic stem cells from progenitors. Very fortunately, Dr. Yokomizo, now he is partially independent, he found that HLF, this is a hepatic leukemic factor, is exclusively in AGM region of the hematopoietic stem cells in AGM region, especially ventral part of the dorsal aorta. So when we use RANKS1 CLE or bubble I CLE, induced CLE, we cannot discriminate HSC from EMP. But HLF is very specific to hematopoietic stem cells. EMP will not express HLF, hepatic leukemic factor. So we analyze the HLF distribution in AGM region. So this is a single cell RNA-seq, and this box shows the hematopoietic endothelial clusters. You can see the endothelial cluster, ellipsoid clusters, or megakaryocyte clusters, and macrophage clusters, very specific. And the HLF expressed in this cluster here. This is very narrow and focused compared with very widely expressing c-KIT. So HLF is a good marker for the hematopoietic stem cells. And we try to trace of the TD tomato positive cells in HLF CLE-ERT mouse. This is the tracking study from E14 to E18 in mouse embryo. So for 4 days, we can see HSC number is very much increased like this. And progenitors are increased like this. However, short-term HSC, like a progenitor, are decreasing in spite of the increasing of HSC. So this is a very interesting phenotype. And we try to screen for HSC specifying factors. So we compare the AGM gene expression in AGM and yolk sac. AGM is a main place for hematopoietic stem cells. So we compare the gene expression in transcription factors. Interestingly, we can see AB1 or GFI1 is highly expressed in caudal half of AGM region, but not in yolk sac. So we try to analyze the AB1 transcription factor. AB1 is very highly expressed in HSC and downregulated in progenitors. And E10 to E14, AB1 is detected like this. So interestingly, heterozygous AB1 knockout mouse. So AB1 plus minus embryo show the decrease of the hematopoietic stem cells or short-term hematopoietic stem cells. On the other hand, we can see the CMP, GMP, MEP, these progenitors are not so decreased. So very specific suppression of the hematopoietic stem cell fraction. On the other hand, when we generated the transgenic mouse of the AB1, we can see stem cell fraction is very much increased. And this fraction can show the engraftment in irradiated mouse. So AB1 is very important for the self-renewal in the hematopoietic stem cells, but not so much in the differentiation of the stem cells. So we can propose this kind of the model. AB1 in the under the very high expression of AB1, we can see a self-renewal hematopoietic stem cells. However, this stem cell will not contribute to the differentiation. Just the short-term HSC progenitors can supply the red cells and white cells. This is quite different from the classical model. Pre-HSC produce HSC, short-term HSC, MPP, everything. And the HSC in this period can show the self-renewal. So this is the implication. Self-renewing process is separated from differentiation process. So one question is it specific for fetal hematopoiesis? However, recently several papers appeared. So this connection between long-term hematopoietic stem cell and short-term hematopoietic progenitors, even under the stress. So for example, very severe infection or chronic infection, we can see the increase of the matured blood cells. However, HSC are not so much changed. That means HSCs are reserved. If this is true, I think ex vivo predominant induction or increase might be possible. We should enhance self-renewal division in vitro. So we studied about cell metabolism in quiescent hematopoietic stem cells. And the quiescent hematopoietic stem cells is very different from cycling stem cells. Mitochondrial membrane potential is low, and they show a very high engraftment rate. And they show autophagy, and protein synthesis is a little bit low. And almost 10 years ago, we have shown that hematopoietic stem cells use glycolysis very much to obtain the energy. This is a not so efficient system to get the energy, but mitochondrial oxidative phosphorylation system is a little bit dangerous because they produce ROS. Reactive oxygen species induce cell differentiation and finally induce exhaustion of the stem cells. So stem cells use a glycolytic pathway and hypoxic bone marrow. So there are so many trials about the stem cell expansion factors. 2010, we can see stem cell regenin. This is SR1 and also UM171 pyrimidindole. We don't know the function so much, but these are increasing the self-renewal of hematopoietic stem cells. And very recently, PVA, polyvinyl alcohol, is a very, very strong factor for stem cell expansion. And very recently, almost 1 month ago, we can see suppression of sphingosine kinase 2 is very important for the self-renewal expansion. Sphingosine kinase 2 is located in the nucleus, and they will induce upregulation of the HIF-1 alpha and glycolytic metabolism. So this is very similar to our previous finding. SPKH2 sphingosine kinase can bind to VHL and Ph.D. So if a knockout of the sphingosine kinase 2, HIF-1 alpha is very much increased. This upregulated HIF-1 induces aerobic glycolysis, and the loss level maintained at very low, and the HSC phenotype is young, and the regenerative potential is very high. So this kind of the approach is very interesting. And also we can identify the EPCR high cells can show the self-renewal. So if we can prepare the EPCR high cells and we can modify the very nice culture system, we can see the stem cell expansion. By contrast, EPCR single positive cell can show the self-renewal and differentiation both. So you can see so many megakaryocyte, CD48 positive megakaryocyte from this fraction. So from now, we have to improve the culture medium for self-renewal system. Especially glutamine or glucose metabolism is very important. Also a scaffold and frequent medium change is important because there are so many differentiation factors secreted from stem cells. And concentration of the cytokines also very important. And oxygen concentration should be regulated. Very recently, we can get these data, EPCR high cells culture, and 7 days, still EPCR, only EPCR high cells are detected. And for more days, we can see EPCR high stem cells like this. So more than 10 days, we can maintain the hematopoietic stem cells ex vivo. So this is a summary. Probability of self-renewing process is governed by intrinsic and extrinsic factors. Some extrinsic factors can enhance self-renewal activity of HSC. And the self-renewing process is separated from differentiation in fetal liver HSC. So ex vivo induction of self-renewal division in HSC might be possible by metabolic modification in the culture. So thank you very much for your attention. And this is a collaboration of the CSI, National University of Singapore, and the Kumamoto University in Japan. And the last one is advertisement. So CSI, NUS, waiting for your apply to this PhD graduate scholarship or PhD fellowship. So Singapore is a quite international city and country. So please actively apply this scholarship. Thank you very much.